In this video, we'll discuss working with GenBank files and look at various GenBank-related data functions and ChemCharts. GenBank files are used extensively in bioinformatics to store information about biological sequences and features of those sequences. A link to the NCBI GenBank webpage with an example GenBank file and an explanation of the contents is in the description. While information-rich and human-readable, GenBank files can be challenging to work with directly. ChemCharts provides various tools to work with GenBank files and uses the GenBank file format to store user-defined information and data function results. For this example, I have a GenBank file containing 300 records with mRNA sequences, each of which has multiple feature entries describing aspects of each sequence. To import this into ChemCharts, select File, Open, and Browse to Local File, then navigate to the folder containing your GenBank file. You can also select it from the recommended list if it's available. Once the file has been imported, you can add a ChemCharts table visual. The data table has 13 columns that correspond to different portions of the GenBank file. The locus line of each record is parsed to give ID, division code, molecule type, sequence length, sequence type, and strand topology. The definition column contains the definition line content from the file, and the source column contains the source line content. Genus and species come from the organism line, and class levels are derived from the remaining entries in the organism block. The GenBank column in the table contains the exact content of the original file, with each row containing each record. At the top of each GenBank cell is the sequence from the origin block of the GenBank record, and this is identical to the content of the sequence column. Annotations in the GenBank cell are derived from the features section of the record. Hovering over the annotations will provide a pop-up with more information about the annotation or region of that sequence. Sequence columns and GenBank columns are treated differently and contain different content in ChemCharts. Some data functions that only require sequence information can use either type, but only the GenBank column type contains the extended feature content of the GenBank record. The content of a GenBank file can be copied or exported in multiple ways. Right-clicking on the table brings up the context menu, and selecting Copy gives the following options. Copying rows will copy all the content of the table to the clipboard as a tab-delimited document. The GenBank column is treated as text and contains only the sequence. Copying the cell will copy the actual GenBank record of the clicked cell to the clipboard. Copying the column will copy the entire column clicked on as text. For the GenBank column, this will copy only the sequences. By choosing Export from the context menu, the entire table can be written to an Excel file or a tab-delimited text file, both of which treat the GenBank column as text with only the sequences. Exported rows can consist of only the marked, only the filtered, or all rows. Columns to include in the output file can be removed from the chosen box by clicking on the X at the right of the column name. Columns can be added from the remaining box by clicking on the plus at the right. The columns can also be reordered in the chosen box by dragging on the tab at the left. There are also options to include column headers and to transpose the table. Choosing to export to GenBank will export a multi-record GenBank file. Specify the GenBank column to export and how to determine which rows to export, again marked, filtered, or all rows. An ID column is optional. If none is selected, the ID from the original GenBank file is used. If an ID column is selected, the entries in that column will be used as the identifiers and replace those in the original GenBank record. Now let's take a look at the GenBank-related data functions. Embed GenBank files provides a way to import multiple single-record GenBank files from specified URLs. Here I have a table with a column that specifies different URLs where GenBank files are found. In the temp directory on my computer, I have 3 standard text GenBank files and the same 3 GenBank files that have been compressed with gzip. ChemCharts handles either uncompressed or gzip compressed files. The table also has 2 valid web URLs that point to GenBank files. In the Embed GenBank Files Data Function Settings dialog, specify the data table and the column containing the URLs. Also provide an output column name. When the data function is finished, the files associated with the URLs are embedded in the table, and a new GenBank column is created. The Extract GenBank Features Data Function will create new sequence or GenBank columns or a new table with rows based upon the feature keys and feature qualifiers of the GenBank file. This data function is particularly useful in combination with the antibody numbering and antibody component analysis data functions. Antibody numbering creates annotations for different regions of antibody sequences as shown here. The different framework and CDR regions are individually annotated and to be extracted to new columns to be used as input for the antibody component analysis data function. In the data function settings dialog, identify the table and the GenBank column. Also specify a column that will provide unique IDs for each row. These will be used as row labels if a table is created by the data function. I'll come back to explain feature keys and feature qualifiers in a moment. Multiple features in a GenBank file may have the same feature key and feature qualifier. Duplicates can be handled either by creating a new table with multiple rows for the duplicated feature names, or by creating multiple columns in the existing table. Output columns can be either sequence columns or GenBank columns, or both. Remember that only GenBank columns contain annotations and feature definitions, and sequence columns will lose this information. But sequence columns can be treated as plain text. Input the maximum number of regions to be extracted. GenBank files can have a lot of feature annotations, and the outputs can quickly add up. Keeping the total number to extract low, especially during development and testing, will make it much easier to debug data function operations. The data function is also limited in the total number of output columns it can generate. The feature key and feature qualifier inputs refer to the feature and qualifier labels in the GenBank file. The GenBank file for this column contains a set of antibody sequences, and the features were generated with the antibody numbering data function. Looking at the GenBank file, we see that there are multiple feature keys called region, and each of these has a qualifier called region name. In the ChemCharts table, each of these regions is illustrated with an annotation. By specifying region and region name for feature key and feature qualifier, the data function will create a new column or a new row for each unique combination of feature key and feature qualifier. Some features may only need the feature key, and the feature qualifier can be left blank. When the data function is finished, either new columns will have been created in the existing table, or a new table will be created depending on the settings to duplicate handling. If a new table is created, click on Configure Visuals to configure the new table. In addition to the sequence or GenBank columns, columns indicating the start and end location of the extracted sequence in the original sequence are also created. The extract sequence from GenBank data function creates a new sequence column from an existing GenBank column. Choose the data table and the GenBank column, and provide an output column name. The data function can also be limited to operate only on marked, filtered, or marked and filtered rows if wanted. When the data function is finished, a new sequence column containing only the sequence from the GenBank file is created. This is useful when you need to treat the GenBank column as a plain text sequence, or you need to create a non-ChemCharts table from the data table. Thanks for watching, and check out our other ChemCharts videos.